pleasure to open the presentation with Anno Kurth from Japan, who will talk about activities related to potentials reinforced place fields by behavioral and scale plasticity.
Yes. Thank you very much, and thank you also for the opportunity to present my work here. Sorry for the very long title. If you read it out loud, you notice that it's actually a bit like long. Maybe this is work I did jointly with Toshitaka Asabuki, under whom I work at Riken in Japan. Okay. So something that fascinates me, like as a neuroscientist, is how the brain, or like we, but like in that sense, the brain can quickly adapt to novel situations, for example, by learning things. So, and I think like this is best exemplified by vision. So this is a specific kind of animal. Most of you, I guess, haven't seen that specific animal yet. It's called a numbat. It's like one of these weird animals that live in Australia, which have like this pouch. And even though you haven't seen this before and you also didn't know the name, probably now you do if I show you another one of those. And the interesting thing now is, how do we actually do this? How can we do like the single or few shot conjunction, other people would call it binding, of different representations in the brain? And yeah, exactly. So if I show you another one, you will be able to name it. And the leading idea here is that this is facilitated by a rapid formation of what is called a joint representation. So like of the visual information and also of the name. And like this is kind of difficult, because if you were only interested in identifying this, you don't need this, right? You don't need to make a completely, you don't need to learn a new representation. This can be done by concept learning and deep neural networks, convolution networks, for example. But if you want to attach the image of an animal to a sound, to the name of this animal, this is more difficult, and this is done with a joint representation, and this is called indexing theory. So how does this work? Which kind of plasticity is involved in the formation of such a representation? And classical plasticity mechanisms most likely are not fully, at least, responsible for this, and this has multiple reasons, because, for example, spike timing dependent plasticity, we heard about this earlier, or homeostatic or structural plasticity, they usually require, under physiological conditions, long periods of time for the consolidation, for the emergence of these new representations. It can work, but like it needs a lot of time. And but if we now look beyond those very classical plasticity mechanisms, what do we actually know about the formation of the rapid formation of new representations? And one example here are place fields. I will come to them in a second, in the hippocampal region CA1. Which are formed by behavioral time scale plasticity, also BTSP for short. So, and because everyone else so far was talking about cortex, and I'm now going to talk about something else, about the hippocampus, I want to remind you briefly where the hippocampus is. So like here we have the nice cortex, you can see it in this like thickness over there, but the hippocampus is like another brain structure. It's like deep inside the brain. It's like quite different architecture-wise from the cortex, and it has a completely different function. And one of this is actually it also has different regions, but like in the sub-region CA1, one function is the formation of place fields. And here we see an example. So if a mouse, or like a rat or a human for that matter, move around a room or like around a linear track, so we will make the simplification that we now consider linear tracks, and we can see that neural activity, this is the activity of a neuron across laps on the track, right? We see that sometimes at some point something happens that already gives it away. It's a CA plateau potential. I will talk about this in a second. But something happens and suddenly we have activity there that is persistent across the laps. So this is facilitated by a large synaptic change that alters the cell responses in a single trial, or maybe sometimes few trials. It depends a bit like on what's going on, but this is like a thing that happens in the brain. So before I want to talk about my work, I want to briefly recap a bit about the physiology and anatomy of like cells in the hippocampus. And as in cortex, actually, we have pyramidal cells in the hippocampus, which, as in cortex, have two kind of integration zones. One distal integration zone. This is innervated by a region EC3, entorhinal cortex layer number 3, which people would claim somehow feed in the current state, something like a contextual input, they would call it, and a integration region which is close to the soma, which is for these pyramidal cells in CA1, innervated by projections from a region called CA3. This carries sensoring modalities, it carries visual information, also carries location. These are, for the people who know, the so-called Schaffer collaterals. Each neuron has like about 20,000 synapses from these Schaffer collaterals. And these are the ones actually that are altered by the behavioral timescale plasticity. So to come back to the place field formation, when a mouse or like a rat or a human, for that matter, enters a new environment, the pyramidal cells in CA1 rapidly form place fields. So, and we have two environments here, environment A and environment B. They are both a linear, like circular track, but they are different. They might have different wallpaper. So the mouse thinks these are actually different things. And what you can see is then that the CA1 place cells, they form like a continuous representation. You have to order, of course, the neuron, right? Like it's not clear which neuron belongs to what, but you can order them so that they tile the space even. If you then, and here you see the activity of one cell. If you then move the mouse from environment A to environment B, and if you keep the original ordering, you won't recognize anything here, right? Like no structure, like no discernible structure. But of course, if you order them again, you will find like this clear representation of the space here. So like this is a general feature, and this is called remapping. And this happens very rapidly, right? And this is also why like this BTSP was hypothesized first, and then like later, of course, also consolidated. So what about this specific kind of plasticity? It's called, as I already said, behavioral timescale plasticity. It is dependent on the presynaptic activity, as we know from SDP, right? Very, very standard. This is usually interpreted as an eligibility trace, and it's also dependent on a calcium spike in the apical dendrite of the neuron. And we can think about it like this. We have the presynaptic activity. This would be here in green, and then like we have the calcium spike, which corresponds to this orange curve here, and the overlap to some extent would give the potentiation of the synapse, which registered the presynaptic spike. So the important thing here is BTSP does not depend, like SDDP, on the postsynaptic activity, right? It depends, has as a go signal the calcium spike. In the following, I will make a simplification and just refer to it as a PP, as a plateau potential, right? Technically, this is not 100% correct, but it's a reasonable approximation. And but as I said, it's not dependent on the postsynaptic activity. So there's a big difference to like standard SDDP rules. And what's also interesting, in contrast to standard SDDP, you have a strong bidirectional change. So both potentiation and depression is possible. It doesn't change the sign, right? Like the synapse is always positive. It's always an excitatory synapse. But depending on the initial weight of a synapse, you can either get an increase or a decrease. And this you can see here, like this would be the initial weight. This is the time difference from the plateau potential. And you can see that if you are around the onset of the plateau potential, you most of the time like get a potentiation. But like if you have the same initial weight, like you get, so if you have the same initial weight and you move, for example, like a bit to the left, like you spike too early or too late, you will get a depression. And also something that is noticeable is the time scale of behavioral timescale plasticity. And this is also why it's called behavioral timescale. It's not milliseconds, like 100 milliseconds as for SDDP, but it's seconds. Like, so there's a, yeah, Mihai, you have a question?
So your y-axis is that initial weight?
Yes.
How does it connect to the RAM, the VM RAM?
Yeah, like if you like stimulate this, like how strong you can, the initial weight alters how quickly the membrane potential on the postsynaptic cell increases.
Yeah, but this is the PSP, not the plateau.
Yes, this is the PSP, not the plateau.
Okay, so it's the RAM on the PSP.
Yes. So like, and this is like when the synapse is already very strong, right? And now offset to the plateau potential by, let's say, 4 seconds, you can get a depression, like if the weight is too strong.
Yes. Now my question is, typically the shortest time constant in the PSP is the synaptic time constant. Rise time is the synaptic time.
Yes.
And that is usually very small, especially for alpha synapses.
Yeah.
Scale of 1 millisecond, or 2 or 3.
Yeah.
Can you really detect changes in the ramp with this scale?
Because, yeah, CA3 neurons, like the input neurons, are bursty. So like you don't only get one input, but you get many.
And so you have a pileup of...
Yes, yeah. Okay. So, but like if the plateau potential onset is important, like for either the potentiation or the depression here, like it's a crucial question, what actually controls the plateau potential onset? And the frequent assumption is that the occurrence is somewhat random. Like if you look at the modeling literature there, they always say it's like, it's a random process. And what we know, however, is that the EC3 input, the input to the apical dendrites, is necessary for eliciting such a plateau potential. So it's tempting to assign this EC3 input, the input to the apical dendrites, the role as a controller of the plateau potentials. But the EC input is highly variable. So this you can see here, this is the input, like this is the activity recorded on one EC3 axon, and like you see it's not really consistent. Maybe it's like consistent if you average over the different trials here. So this is like over time, one axon over many trials, you could see like a peak here and a peak over there, but like it's not really that consistent. And also if you look at it, and this is just a single axon, so if you now average over many axons, you might say it's consistent, but like generally it's assumed it's not very consistent. Yet subsequent plateau potentials tend to cluster at close to the location of a previous one. So here you see the place field of a cell in CA1 and the running lab, and you can see how this place field, how the preferred location drifts, right? It first drifts a bit in this direction, then drifts back and so on, because this is like the space. This one is a bit more stable. It only drifts slightly to the left. And this means if we now assume that each of these changes is triggered by a plateau potential, that the plateau potential is fairly like close to where previously at the location a plateau potential happened, right? Like, and this is a bit of a conundrum if you know the high variability of this one. So, of course, I'm not the first one to identify that there's somewhat of a tension here. And like usually people would now say, yeah, this is target learning. A bit glossing over actually the unreliability of the EC3 input. Like what these experimenters suggested, and these people here, Guillaume Clopart, model, is that you get the target. The target is actually the EC3 input. This is the target. And then the cell, the CA1 cell, wants to learn this input by adjusting its input to the basal dendrites. In order to then actually learn something, or like to know when learning is done, you need to somehow feed back the CA1 activity to say, actually, I don't need to learn anymore. I learned already everything, so that the plateau potential probability goes down. But this, as I said, doesn't really fit to the high variability of the EC3 input. And also, this is like a rather like peculiar architecture. Like you somehow need to assume that a neuron innervates another cell that then innervates back to that cell. This is what Guillaume Clopart assumed, but I find it a bit of a stretch, to be honest. Yes, and like here you see again why like they believe this, where you see the EC3 input, which remains fairly constant, but over the time, the correlation between CA1 and EC3 increase. So like this is why they say this is. But I have a different idea, and like this is what I want to talk about. I want to computationally study, and this is like where my own work starts here, study behavior times spatial plasticity for CA1 place field formation. I want to especially address the question, what controls the timing? Is it, for example, a higher order signal as in the target learning framework? Is it a local trigger? Is it maybe a combination of both? And I want to propose to you a unified mechanism for the initial and recurring onset of plateau potentials for behavior timescale plasticity. The idea here explores a variation of a theme by Larkham that recently got some additional actually experimental support. These people here showed that if you have a CA1 cell, 3 somatic spikes in quick succession lead to a so-called dendritic spike. So what is a dendritic spike? If you have a if you have an action potential at the soma, we know that the action potential travels back. It travels back to the apical dendrite. And usually it's heavily attenuated on its way down, right? You can really see here it's very big, here it's very small. But sometimes when we have a dendritic spike, this is what they call D spike here, you see that it's actually amplified, relatively at least, on its way down. And yes. It's, what's the element in English? Potassium channels. Is it K? Is K potassium?
Yes.
Yeah, it's inactivation of potassium channels. Yeah. I know, and so and I mean, and there's also some sodium going on, of course. Like, but I mean, there's like it's really complicated to figure this out. They're actually a very nice model. So, well, I mean, this is hippocampus, and hippocampus the pyramidal neurons are the other way around. So, like, this is bottom. So hippocampal, like, so I showed you a wrong side, like for hippocampus are wrong-sided earlier, but like they are the other way around in hippocampus.
What I'm asking is that in the standard argument have NMDA potentials actually interacting with that propagating action. So the dendritic stuff is NMDA driven.
Yes.
But that is different from what you're
Yes, this is different from what I'm going to say. And I mean, this is like very cool paper. Like we can talk about this later again. But yeah. So, but I mean, sodium is of course also important. Like it depends on inactivation of various channels that then boost this going back. So this is pretty cool. And so conversely, also if you have a lot of somatic spikes, these gates close, and this mechanism is disabled. You're not able to have another. Like they need a cooldown then. And what they also then, to some extent, show, more hypothesized, I would say, is that plateau potentials result from a dendritic spike and coincident EC3 input. Right? So this is what they then what they claim, and this is something I want to now investigate a bit more. Yes. Yeah.
Okay. What I know about the dendritic spikes, that if we have a lot of EPSPs that happened before, so the back propagation of the action potential should be lower, not higher.
If we have a lot of them.
Yeah, if we have a lot of them, like in
yes.
small time.
Yes, yeah, this is like basically this. So elongated periods of activity disable dendritic spikes. But if you had a period of relative silence, and then quick spikes, three, 4 spikes, neuron dependent, let's say 3. No, if it's an onset of 3 spikes, it enables it. Like then it boosts it. So like there's a somewhat of a flip. If the neuron was silent and like you have 3 spikes, then you have a boost. But if the neuron was active in a high state for a very long time, it's disabled. Yeah.
And can these dendritic spikes be triggered by presynaptic or only by subsequent reference?
Sorry?
So these dendritic spikes, can they be also triggered presynaptically or only through back propagation?
In principle, they can also be triggered presynaptically. But like, I mean, it depends. Is the input strong enough? Is the input coincident enough? Right? Like, yeah. Okay. So what I want now to explore is a unified mechanism for the BTSP inducing plateau potential onset. My claim is it's from input fluctuation drive quick succession of somatic spikes when the neuron is silent. And this triggers a dendritic spike that makes the cell plateau potential ready. And if I then have enough coincident EC input, EC3 input, we generate a plateau potential. So this is basically what I want to do, and I want to validate this idea, this approach, with 3 kind of experiments where I want to show that this can, to some extent, successfully explain mapping, remapping, and representation drift in CA1 cells, EC3 dependent overrepresentation, and cross-day reinforcement of place fields. Okay, so my model, my neural model here looks as follows. I have a 2-compartment neural model. I have an upper compartment which gets EC3 input. I have like my somatic compartment which gets the CA3 input. I have a membrane potential which is just the sum of PSPs with the weight, so that's a linear neural model. And then I choose a spike response model here. So like I have a nonlinearity, and then I have to sample some somatic spikes here from that, right? So some people I know don't like spike response models. This also works with a hard threshold model. So I already tested this, but I mean, this is for simulations a more convenient framework. So I record the spikes for 200 milliseconds. This is the time scale I get from the data. And if I have more than 3 somatic spikes in 200 milliseconds, I trigger a dendritic spike, provided that the neuron previously was relatively silent, where I have a similar mechanism where I have to go 500 milliseconds in the past and count the spikes here. It's basically like a ring buffer. buffer we have where I have to register the spikes. And if a dendritic spike occurs, this opens for 100 milliseconds a window for plateau potential generation. So these numbers here are like biophysiologically constrained. This means I get my EC3 input, like it wobbles around like this, and I get a D spike, it opens a window, and then I basically mask this, and I can take this as a plateau potential rate over here. Does make sense? Yeah, okay, good. Then upon the plateau potential onset, I calculate the time difference between the plateau potential onset and the presynaptic spiking activity. Like this is very similar to SDDP, where you take the postsynaptic activity, but I take the plateau potential onset basically. And then I change the weights from CA3 to CA1. And you can see here, this is my plasticity kernel. As I said previously, my plasticity kernel depends on the relative initial weight. If the relative initial weight is very low, I get a strong potentiation. And if the initial weight is already high, I can go down to a depression. But the cells can never flip, or sorry, the synapses can never flip the sign, right? An excitatory neuron synapse can never be inhibitory. This, by the way, already implements something like an upper cap for the weight of a synapse, right? Like, because if it's too strong, like it cannot be potentiated any further. So the network model then looks as follows. I have like multiple of these pyramidal cells. They are recurrently, there is a lateral inhibition here. The lateral inhibition is necessary to spread out the place fields nicely so that you don't get an overrepresentation of one particular location. And then you get the EC3 input. It's at first a stationary process. If I deviate from this, I will tell you. You get spatially tuned CA3 input, right? So this is also what CA3 activity looks like. And we have all-to-all weights, which are random, like random positive weights here. Like, so there's a bit of variability here, but not too much. Okay, so if I do this, if I simulate this kind of thing, a mouse runs down like a track, you have CA3 activity, which somehow tracks the location of the mouse. You can see that sometimes if enough spikes occur, you get a plateau potential. This would be the first one here, and you start forming a place field, right? But because here it's not very consolidated yet, you can get like other plateau potentials at different locations. And then again, right? So like this is a known phenomenon. This is called translocation. It sometimes happens, but usually what happens after a couple of steps is that the neuron, this is again is the labs here, figures out, it establishes a somewhat stable location. There's always some drift, right? Usually if you would simulate for longer, or like if you would do the experiment for longer, there are like other plateau potentials that move this further to the side, but like it's fairly stable. The population activity, as observed in the experiment, evenly tiles the space here. And this is just like, for example, how the EC input looks like. It's like, just as I said, a stationary random process. So like there should be no relationship between this, and there's actually not. Yes.
This developing in such a sort.
Yes, yes. I can tell you. The reason for this is that initially the plateau potentials are kind of random, because like they come from like a random, like from a random somatic spiking, which triggers this. And then you have a form of lateral inhibition here, which like then evenly spreads this out, right? So this is like the idea, because if I'm like, I spike, I have a place field, I'm very active, right? And now I inhibit you, and now you're not so likely to have a plateau potential yourself, but at another location. Okay. So this, we can see it clusters around here, and this is the EC input is actually constrained by some experiment later. Let's look at one place field. This has a very, it travels a bit more. It has a different kind of place field evolution. This is the final place field, and these are the final weights if I sort my input neurons accordingly. And now I will look over time at the evolution of these weights here. So the weights that correspond to this ordering, to these, and so on. And you see, of course, at the rightmost rim, like nothing happens. There's never activity here. The weights stay largely unchanged. If I move one to the left, so to these here, again, not much happens. But now here, for these weights, of course, initially there is some activity here. But over time, this gets depressed again because the cell travels further to the right. And we have like this depressive part of the BTSP. And so why it travels in my model, or like why it travels in general? Okay, why it travels in my model is because you have, of course, if you look for inactivity and then 3 spikes to trigger a plateau potential, this will tend to happen at the rise of the activity, right? But this is like a general feature observed actually in the data. Like, so this is something you observe, and you can only explain the drift in these cells with the BTSP. So there was like some work showing this. Because like you usually move from location, you move in this direction, right? Like from a starting point and then you move. But it doesn't always travel to the left. So for example, this plateau potential here is fairly to the end, right? Because the cell also was not so active yet. Okay. So, and we can.
Why is the thing straight?
What way is the
Yeah, when's the drift? Where does the drift come from?
The drift comes from that if I run again, so I run multiple laps of the same trial, and then I run and I have a place field, right? And now the neuron fires more at the onset of the place field. So the place field looks like this, right? So I move down here, so I have this firing rate. So as the firing rate increases, there is a higher probability to have like these 3 spikes in quick succession, which triggers the dendritic spike, which triggers maybe the plateau potential.
But it cannot move far to the left, presumably, because there's another neuron already.
Yes.
There.
Yes. Yes. Yeah. Correct. Okay. So, and if we now look at the weight evolution, for example, now this corresponds to this part here. They always are like kind of high, and so on. So these are all the weights. The drift has a consistent bias that gradually decorrelates the place field representation. This is like population activity at lab 10. This is population activity at lab 60, sorted the same way. If I would sort this new, it would look again like this, right? Like, so there's really like some, exactly, I can sort it always to look nice, but you see like there's a persistent drift in this direction. And what this leads to is that it leads to a, if you compare the unsorted activity, it leads to a like gradual decorrelation of like your representations here. And this shift here to the left can be explained by the fact that the location of the plateau potential is like rather close to 0. I mean, it's like that, it's not completely random, it's like rather close to 0. It usually happens around where previously plateau potential happened, but not exactly at the same location. But this gradual decorrelation is actually something you observe in experimental data, where if you let a mouse run in environment A0 and then environment B10, B70, so for 1, 2, 3, 4 times in the same environment B, you can also see here the same decorrelation over time. Yeah, so like this aspect is consistent with experimental data. So if you now move to another environment, what happens actually then? Like what triggers the remapping in CA1? And what triggers the remapping is the input changes, right? So we know that the same cell in CA3 that in one environment, say, codes for location 1, in another environment can code for location 10. So like you have a shuffle down there. So already on the level of CA3, there is a remapping which is then transferred to CA1. And what I did then is I simulated my computer mouse here, like in environment month for a certain amount of time. Then I changed the environment, and then I had the original environment again. And I'm interested at this time point at lab 20, at lab 40, and at lab 41, denoted here by these symbols. And if I now look at the activity, I can sort it according to this time point, so get a beautiful activity here. In my novel environment, I get a complete decorrelation, but of course, if I now look at the original environment again, a certain structure still remains. Weights are destroyed, right? Weights are destroyed because there is the depressive part of BTSP, but not completely. And I get a bit of surviving input. And the same, of course, holds true if I sort according to this, that I will still see something here. And if I sort according to the new environment, this will, of course, give a nice representation of the new environment, but like the environment 1 will be completely scrambled. So this is also like how this actually looks in the data, right? So this is what happens in the data. And if I now look at the correlation between lab 20 as a as a function of lab, right? So you have a correlation 1 at lab 20 because this is my reference. Then you can see a steady decorrelation over time. This is what I already showed you. But now these black periods, they are the time spent in the new environment, right? And if you look at this, that this year and this year and this year, this is like after re-entering my old environment. And these values tend to be larger than these values over there. So if you spend time in a new environment, the decorrelation is slower, as you would expect. So this is not crazy, so this is kind of expected, but you can also see it here, that these points here, they correspond to the first time point in the old environment again, and you can see that this decorrelates slower than the drift decorrelation, like then this baseline over here, which is also consistent with experimental data, and this can be explained, or this is facilitated by the retention of certain weights, right? Like certain weights are just not depotentiated by the BTSP because like of a configuration of the input. So this is kind of clear. Okay, so this was the first part about like drift and remapping. But there's another thing. There's also EC-dependent overrepresentation, because up to this point EC was stationary, and this is a good assumption if there's no reward in the environment. But if there's a reward in the environment, you get an increase in EC activity. And this increase in EC activity will lead to an overrepresentation in the place fields. This you can see here. You have a certain slope here, then you have another slope here, and then you go back to your original slope, right? So like this is normal. This is an overrepresentation of the location here. You have more cells in here, and then you have fewer cells again. This goes away if you, and this is what happens here, if you optogenetically inhibit the EC3 exons. So if you do this, the overrepresentation goes away. You can see this here in this TD tomato. You see a peak of the representation around the location here, and otherwise it's like flat, I would say, like somewhat flat. And yeah, yes.
I didn't see what did you mean by that?
So you have a slope here,
Yeah.
then you have a steeper slope, and then you have a shallow slope again. And the steeper slope means that, so this part and this part, like, but this is a very big part. This is called the overrepresentation, right? Like you have more cells at this location. And if I now change my EC3 input, and it looks like this is according to data. So like it's a model that is constrained by EC recordings. So like this looks crazy, but it actually seems to be the case like this in the EC input in the brain. You can also get a very similar overrepresentation in my model, right? So like this is basically what happens then here. And then if you compare the with reward and without reward, population average activity as a function of the location, and the without reward, it's like fairly flat, and then with a reward, you have like overrepresentation. This is in the model explained by a higher plateau rate around the reward, right? Like this is what you would expect. And this is also what you measure, right? That the plateau rate is higher around the position of the reward. Okay. So to come to my final point here, and this is cross-day reinforcement, because this is the last effect I'm going to talk about. And single cells can have consistent place fields between days. So this is a bit contradictory, right? Like previously I told you it decorrelates heavily. If you're for a long time in the same environment, like your place map decorrelates. And now I'm telling you something about the same place field between days. And the reason why this is not a contradiction is because these measurements, you have like relatively few expositions in one environment on one day, right? So like, because usually people don't let their mouse run for 5 hours in the same environment, right? Like usually we just measure for 20 minutes and then the mouse is done for the day. And you see this clear decorrelation only if you like really measure for like very, very long time. So this is not a contradiction here. So, but what you can see is that over the days, some place fields, so this is the location here, they are like fairly consistent. They're just here. And you can, for example, see here at day number 3, there was very little activity at the beginning, and then you get a high activity again. While for other cells, they're only active like for 1 day even, or like maybe for 2 days, but then they die out again. So this is like a phenomenon you observe. And what the Vaidya et al. Showed is that place fields are not conserved fully and partially only, and there's only a weak retention of the activity on the next day, even for the cells that are conserved, but they are reinforced across days with BTSP. And so what I then do to represent this in my model is I introduce days. So I end like my simulation after 20 laps in an environment. I use EC as a selector of neurons. So like I have a big pool of neurons now, and I say like, you 100 neurons are allowed to have a plateau potentials and the rest not. And then the next day, another set of 100 neurons can have a plateau potential. And at the end of each day, there's a partial decay of the weights, right? So like the weights decay, this is presumably true by the mouse being in a different environment and having a depressive effect. Maybe the weights just decay back to a baseline anyway. So this we don't know. And if I do this, my what you can look at in the Vidya data is what they show is that the number of place fields across days for a given environment stays fairly constant. These are the new place fields. So the place fields that were not already present on the previous day. Of course, at the beginning, all place fields are new. Then on the second day, not all are new anymore because like you have a part that is retained and this decays here and at some point this crosses here. And this is basically how many cells get newly added each day. At the beginning, all of them. Then at the second day, like this is the ones that survived. These are newly added. Then at the third day, these and these already survived and they are added and so on and so on. And I qualitatively can also see this, of course, in my model because the mechanism is as such as if I already have like somewhat of an activity there, not the full activity, but somewhat of an activity, I have a higher probability to redo my plateau potential at that specific location. Okay. So, and with this, I want to come to the conclusion. I suggested here a unified neuron-controlled mechanism for PP initiation. The idea here is that the activity of the neuron to some extent can control the plateau potential onset. I showed that the mechanism is consistent with experimental results regarding the place field formation and drift. It can explain a place field overrepresentation and also dynamics of cross-day place field reinforcement. And thereby the model provides a like unsupervised mechanism for rapid formation of representations and their maintenance. And how does it actually connect to what I said at the beginning, right? Like, okay, so actually this indexing of numbered and of the image of numbered and the word numbered happens in the hippocampus. It's not a cortical thing. We know this because if we have patients with hippocampal damage, they can't do this. They can't do this in a one-shot way. They can do this over a long period of time. If you sit there the entire day and say numbered and you show the image, and then you say numbered and you show the image, then they can learn this, but not in a one-shot way. So like hippocampus seems to be important for this, and this kind of plasticity seems to be very helpful for this because it's exactly one shot. And this is what I want to say. It's actually, it may involve in this binding or conjunction, but, and this is more interesting now for me even, BTSP-like plasticity, by which I mean a plateau potential as an instruction signal and rapid changes in feed-forward weights, have been also observed very recently in pyramidal cells in the cortex. So like it doesn't seem to be only a mechanism in the hippocampus, but it might be also cortical.
Where?
V1, V2. Yeah, like in also the most crazy areas, like V1, V2, what? Okay, but this is very interesting for me because like previously, as also Marcus said yesterday, I worked on cortical models and I would like love to explore what consequences, how this generalizes and what consequences this kind of plasticity and maybe such a mechanism as I presented you here today, right, which fits to Larkam's theory, and what are the implications for cortical processing. And with this, I want to thank you for your attention.
10 minutes.
Yeah, I try to be on time.
Which are not measured.
Yes, I mean, this is what I'm currently like trying to do. Yeah, so like the first step was, of course, like reproducing the known results, whether it, like somehow is consistent with this, and like then the next idea would to try to make some predictions. Yeah, but it's like not so super easy. But I mean, I think like this is usually the crux with modeling in computational neuroscience, right? Like that making the predictions is kind of difficult. Yes, sorry, you had a question?
We try to analyze the differences in the direction of the movement along the circle.
Yeah. So what I, of course, in like my simulation, as is like common in the field, usually like what you simulate is like one continuous same speed movement in one direction. But of course, like if you stay at some point, this can already lead to an over-representation. So like for a long time, people said over-representations come from the fact that the mouse runs to a reward zone and then stays at the reward zone. And then if you assume like you have an identical rate for the plateau potential onset, just by the virtue of being longer at one location and going a bit back and going a bit forward again, right, you will have more higher over-representation. But this was like to some extent invalidated by this optogenetic kind of experiment. Like when you shut off this EC3 input, you don't get this over-representation. But like, yes, yeah. Yes, Marcus.
You didn't say how large the network models are.
So like, currently they're fairly small. Like currently it's like 1,000 neurons. Yeah.
You map all this back from neurons.
Yeah. No. I mean, it's also not like completely unrealistic number, but of course, like there are effects you cannot capture with such a small model. Yeah. If not,
I think there is this famous hippocampus research.
yeah, Tom McHugh, yeah.
Does he know about this?
Yeah, I've interacted with him. Yeah, he's a good guy, nice guy.
Questions? No. Thank you, Asta.
Thank you.
And the next